Well, I hope all you students are enjoying this as much as I think all of we more experienced people are enjoying it. It's wonderful to be with so many people and hear about so many different techniques and having them be brought up to date for us right now. So thank you very much for including me in this. Okay, I'm going to talk about our long-term study of the right whales at Peninsula Valdes, Argentina. It's our study area is about the same latitude we're at right now, but it's just on the eastern coast. You can see it here jutting out into the South Atlantic Ocean, and it's a nursery ground for right whales. Our study began in 1970, when Roger Payne, he'd worked a bit on humpback whales, and he wanted to see some right whales and was told, I think there's some at Peninsula Valdes. So he went to the cliffs of the peninsula and looked at the whales, and he realized that he could tell individuals apart from the white markings on their heads. And at that time, it was the time when there was a transition between learning about whales through the whaling industry and starting to study living whales. And Roger realized that if he could tell individuals apart, he could follow the same individuals throughout their lives and learn an awful lot more about their biology than was currently being learned. So what he saw were these white marks on the heads of the whales. They have them in the same place that humans have facial hair, so they have them above the eyes, like eyebrows, along the lower jaw, and then between the blowholes and the tip of the snout. So it's like mustache, it's like facial hair. And you can see in these two whales that this whale has 1, 2, 3, 4 patterns, and this whale has a kind of an L pattern. So he recognized he could tell them apart and started to study. What makes the patterns white are not that the skin tissue is white, but they're covered by thousands of amphipod crustacean or whale lice. And one of the things, if you want to do a study like this, if you're a student and thinking of doing this, you have to make sure that your marks don't change over time. So we luckily have some whales with white back marks that don't change over time, and we had repeated sightings of those whales over years and found that they stay the same, though these whale lice, cyamids, can crawl all over. I've often thought in the middle of all that, what must it sound like? It's kind of like Grand Central Station with all the whale lice talking to each other, but they're restricted to the callosities, that species, so it stays on the head. So the technique that Roger developed for studying the whales was to make aerial surveys of all the whales that were the peninsula, and he rented a small plane, a Cessna, and flew along the perimeter of the peninsula, and every time he came upon a whale, he photographed it, he recorded its location, the companions that were with it, and particularly noted whether they had a calf or not. That was in 1970. Here's our current John Atkinson doing one of the aerial surveys. We've also used a number of techniques at Peninsula Valdes because it's one of the few places in the world where you can study the whales from land, which is fabulous. So this is our observatory here on the top of a 60-meter cliff, and the whales, when they're in the area, stay along the 5-meter depth contour, so they just pass beneath you and you take photographs and observe what they're doing. But as a result, the data that I'll be presenting are things that Roger found and discovered, and then a string of graduate students and people that have worked with us who began, I think they teased on whales working at Peninsula Valdes at that point. So the cliffs are great. You sit there and there's the water beneath your feet, and here's the kind of view you get of a whale. And it'll take an hour and a half for a mother-calf pair to pass by, often if they're doing resting like this or gently playing. So you can record a lot of things from the cliffs about the whales. A lot of important things. So this is, rather than going from the grand picture that John just gave us, this is like going through the magnifying glass and what you can do in your own backyard if you're lucky enough to do this or have whales nearby. So we use aerial surveys to document the whales in the area, and then from the cliff top, Roger used a surveyor's theodolite to track the movements of whales, so you can determine things like their swimming speeds and individual movements, how they respond to boat approaches. Peter Thomas got his PhD doing focal animal follows. The whales are at Peninsula in the nursery season, and as the calves develop, they go through 3 different, very distinct developmental stages, which the vulnerability of the whales in that time probably changes. We've also done, you can do behavioral, to study behavior patterns, so you can understand what's the normal behavior in this location, or this for this age of animal. Then Chris Clark got his PhD using hydrophones to kind of to record the sounds the whales were making, and then you could follow individuals and see how they responded to when one whale made a whoop, hello sound, then what did the other whales do. And then more recently I've been doing focal animal follows to document blow intervals as a way to assess the whale's body condition. If you have to breathe a whole lot more, you're probably in poor body condition than another mother who can hold her breath for longer. So when you get a catalog, and now our catalog has about 2,800 whales in it, and if you wanted to compare it to other catalogs around the world, you eventually have to get a computer identification system, and this is the system that Lex Heavey and Phil Lovell developed for right whales that are used in Australia and South Africa and at Peninsula Valdes. And what it does is it takes the 3-dimensional pattern on the whale's head and converts it to a 2-dimensional image, so that a photograph taken of a whale from any angle can be compared. And what happens is the program lines up the closity patterns, and if they don't quite match, it counts how many pixels off is one island from another, and it goes through the whole catalog of these 2-dimensional abstracts. And then lines up all the whales in order of which is the best match to the worst match. So the catalog is always reorganized according to how well it matches. The fun part of this is you get to color in the closities, and it's just plain fun to do. So we found from doing the comparisons with catalogs that you can determine the kind of range of the animals you're studying. And so whales from Peninsula Valdes have been found on another nursery ground off southern Brazil. Females are having calves at Peninsula Valdes. In some years they go to Brazil, but also it seems off Brazil there are many whales there that don't come to Peninsula Valdes. Also, we had 4 matches with Bird Island in South Georgia. People send you photographs if they're in these areas of whales, and hopefully we can get photographs through the IWC to find out where they're going in places that are inaccessible to us. And also 1 male was found here, and a female from Peter Best's population off South Africa was found here, so it can tell about the whale's long-range movements. The basic things that we found initially were trying to develop population parameters, so we could estimate the size of the population, whether it was growing. And so we found out that females have calves once every 3 years, and they tend not to return in the year when they don't have calves. If we'd just been counting the whales and not following known individuals, we would have flown over and counted how many females had calves in a year, and it would kind of look the same from year to year, but we'd only be counting 1/3 of the population. So following known individuals is a really important technique to use. So our longest, now it's 40 years later since Roger started the study, our longest reproductive history is whale 143, who's had 11 calves over that time, over a 36-year period. This is whale 1371, and here she is. We have long reproductive histories of the individual whales. So here's 1371 when she was a calf, and then she was sighted with a calf of her own in 1984. And then she was sighted in 1994. You can see her closity pattern has this long island, 2 islands, and then a kind of hook to the right. And here she is in 94, and here she is in 2008. So those are 3 of her calves, but I think she's had more than that. These were the pictures, the best to show you. And so from the repeated sightings of known individuals, we found that the calves, the age of first calving the mean age is at about 9 years old, though some whales were first seen with calves at 7 years. And also that the mean interval for calves, the number of years between calves, is 3 years, but they also have 4 and 5 year calving intervals. Those intervals are really important because they indicate that the females lost their calves. They have a calf, they're pregnant for a year, they lactate for a year, and then they spend a year recovering. And so if you have a 4-year or 5-year calving interval, it probably means you lost a previous calf, and it indicates the health of the population. And when you collect this data, you may not be population modelers, but there are many wonderful people who are, like Justin Cook, who does our models, or Jeff as well. Anyway, so we've determined that our population was growing up to the year 2000 at a rate of 6.9% a year, and it's around that for all the southern hemisphere right whale populations. But Justin in his model also, this is a graph showing you expect whales to have calves once every 3 years, but in some, and so if you have a calf in 71, you expect it to come back in 74 and in 76. But if it came back in 75, it didn't come back in 74, and you would have fewer calves than expected. So there were certain times, and this is when the database had 30 years long, when the females had this. If you hit the 0 line, that's the number of calves that were expected in this year. In these years, they all had more calves than expected, and here they had fewer calves than they're expected. Oh, sorry. And these years is in 1983. And again, here and here, these are El Nino years when they had fewer calves than expected. In those same years at South Georgia, which is a place where we think the whales feed, it's the richest krill abundance in the Southern Ocean, the Gentoo penguins and the fur seals had lost chicks and pups. So and so then Justin worked with Russell Leeper, and Russell kind of looked to see if there was a relationship with these El Nino events. And it turns out when the waters around South Georgia are warmer than usual, the sea surface temperatures are higher, there are fewer calves born. There's a significant relationship between the water temperature around South Georgia, and when it's cold, there are more calves than expected, indicating that it probably was krill abundance that caused these mothers to lose their calves in these years. But in recent years, since 2005, we've been suffering really high calf mortalities at Peninsula Valdes. We don't know what's going on. Last year in March, there was an IWC meeting to consider what's happening. Why are we having so many calves die? Usually we have 20 dead whales in a year, and we had 482 whales die. 89% of them were calves. In their first 3 months of life, newly born calves. So at the meeting, we presented all our evidence, and the conclusion was, well, it could be disease, but we've done necropsies on all the whales in good enough body condition, and we've found no common disease in any of the samples that have been analyzed, so it seems unlikely it's disease. It could be low food abundance, low krill abundance, so we need to look at that. And also it could be, let's see, biotoxins, toxic algal blooms, shellfish poisoning. Right whales eat copepods and krill, but at Peninsula Valdes they primarily eat copepods. Copepods eat diatoms, and the whales start feeding at Peninsula Valdes in October on spring plankton bloom, so that's a possibility. So with Russell's initial finding about the whales responding to low krill abundance, we've been kind of pointing our current research to trying to figure out more about where the whale's going to feed, what is the krill abundance, and in investigations in the dead calves to look at blubber to see if we can determine was it copepods or krill, what have they been eating. So we've been using stable isotopes to kind of follow migratory paths. We'd love to follow them up do satellite tagging. But if you use stable isotopes, carbon isotopes change with latitude. They're lower at higher latitudes and higher. So if the whales go south, you expect to have lower carbon isotopes. And if they're feeding on the Patagonia shelf, it's higher. You can see these are copepods and krill. And on the Patagonian shelf, they're all green and yellow, indicating they're all kind of higher in this category, whereas this is the polar front, and south of the polar front, they're in these colors, so they're lower. Their prey is, when an animal eats something, the carbon isotopes in the food gets consumed, and it turns into your hair. So with the whales, it turns into their baleen. And a right whale baleen plate, Here it is from the young Luciano to older person. And we sampled the baleen plate at 2 centimeter intervals all along the plate. And this, the blue, is the carbon isotope range. And down here, these are the lower isotope values that indicate feeding on South Georgia. Up here are the values that correlate with feeding on the Patagonian shelf. This is where the whales die, and we've had 5 adult baleen plates, and this is the level of death, and that's the signal at Peninsula Valdes. It's a fasting signature. They're fasting for those 3 months, so they're living off their blubber reserves, which are a mixture of the copepods they ate here and the krill there, so it's something in the middle. So this gives us a 6-year record of where this whale fed, and you can see at the end here it became sick and died. Its foraging record is strange, different. Oh, sorry, and this is the gum, so this is the most recent where it died, and this is the older two. No. So when I overlay the isotopic signatures of their prey, this is a Patagonia shelf signature here, and this is the Antarctic Peninsula and South Georgia. And you see all these dark values are lower, so it down in this here, this indicates they were feeding on around South Georgia in the southern part of their range, and here in the northern part of the range. This is a whale that we knew its age when it died. You don't often know that, but in 1983, when the El Nino event occurred, you can see here, if it could have gone down here looking for food, but it just has a high signature, whereas in this year it did feed down here, and then it fed at Peninsula Valdes, then it came down, so it looked like it didn't have much of krill in its diet in that year. And what we're wondering with the baby baleen, if baleen develops in the calf's mouth when if they're like other mammals, it develops when you're halfway through gestation. So that would put the whales, the baby baleen would be developing in utero when the mothers are down in this signal. So if we can look in the 300 baby baleen samples we have, if it shows a signal down there, it indicates they could have been eating or not eating down there. If it was a higher signature, then that would mean that there was low krill abundance. Am I done? 2 more? Okay, good. So I'll just go on. This is one of our partial albino whales. I'm going to rapidly, you should talk to Luciano about this. This is his doctoral thesis just recently, where he studied isotopes and the genetics in biopsy samples from living whales. And among those, this would be equivalent to 1 baleen plate where whales fed down low and they fed up high. So they fed on copepods and down low, except it's his 131 mothers. So that was an instantaneous signal from the skin of the whale as opposed to the baleen of the whale. So this indicates that all the mothers that were feeding had previously fed over many different parts in different locations in latitude. And then he haplotyped all those whales. These are the haplotypes he found. And when he combined the two, like for each haplotype, this is the range of their isotopes with carbon and nitrogen isotopes. He found that haplotypes have particular signatures indicating that the calves are inheriting their foraging areas from their mothers, which then raises the question, if there's no low krill at South Georgia, can you learn to feed in another location, or does one family line die out? I think that's it. Yeah, sorry. So I won't go into the die-offs. They're dying, and I'll talk about them later in the population modeling section. Eugene Murphy from the British Antarctic Survey proposed that if the sea surface temperatures in the Scotia Sea, which is the area for the richest abundance of krill, if it increases by 1 degree, the Antarctic krill productivity will be reduced by 95%. So for all these whales, if it's true, I don't know what's going to happen to the Patagonian right whales if they're krill dependent, and so that's one of our worries about climate change. Thank you.